Dr. Nadav Ahituv is coming. I'm sure he could say that better than me with his accent, but I did my best. So he's going to be talking to us about a new approach that you all haven't heard about before. And this is using CRISPR activation, or CRISPRa, to upregulate existing gene copies as a novel therapeutic for the deletion genotype of Angelman syndrome. He comes to us from the University of California, San Francisco. I actually was on a panel with him giving a talk, and he gave an amazing talk. And I thought to myself, who is this guy, and how do we get him to work on Angelman syndrome? So I came up to him at the end, and I was like, I think what you do can work on Angelman syndrome. And he's like, I think you're right. And so here we are, about 9 months later, and we are very excited that we are funding a grant for Nadav to be able to work on this very exciting strategy. So I'm going to leave it to you to share with them all of the details. You have a mic, this is advanced, and that's laser.
Great. Thank you so much. It's really an honor to be here. As Allison mentioned, I'm new to the Angelman community, so looking forward to learn more. I've learned a lot yesterday from the meeting, and please reach out to me and talk to me after also. We're very new also to the research side and the science side, so very happy and very honored to be part of that. What I thought I'd do today is, and start with disclosures, of course, is first introduce myself since we're new to the community, and then talk about gene regulatory elements, which is what we work on, and show how they can be used for therapy, and then end in what we're thinking of how we can help in terms of Angelman syndrome. So what you see here is actually a selfie of me. When I was 13, I was diagnosed with scoliosis, and I got a present of this brace that made me socially lovable until I was 16, and at 16, the brace didn't help, and I had spinal fusion surgery that you can see here. I have 8 fused vertebrates, and not as athletic as most people. This really got me into genetics and genetic disease. Both my sisters also have scoliosis, and really learning and understanding genetic disease, but not only just understanding it, but going all the way to therapy. And that's been always the motivation in my life and the thing that really has been driving me in my research. I started my lab 16 years ago at UCSF. I also direct the genetics now at UCSF, and all my work, as you'll see, has been on regulatory elements. So what I thought I'd do first is just explain what gene regulatory elements are. And so you can think of the nucleus of where our DNA is as something like this, actually, where the genes are sort of these metal plates, and to them connect a lot of wires that basically tell the genes when to turn on at the right location, at the right amount, and at the right time. And actually in our genome, only 2% of our genome is made up of genes. 98% of our genomes is not coding for protein, it's not genes. And those are these regulatory elements that tell the genes when to turn on and when to turn off. We in the lab are particularly interested in gene regulatory elements that are activators. And so if you have a gene, and you can envision a gene as sort of a light bulb that should turn on in the right location, in the right time, and in the right amount, what this light bulb has always next to the gene is what's called a promoter. And that promoter is sort of a simple on-off switch. It just turns on that light bulb. But that's not enough. In addition to that, what you have are what's called enhancers that are sort of the dimmer switches that tell that promoter when to turn on, at what levels to turn on, and at what timing to turn on. So they're sort of the dimmer switches of that promoter. And mutations in these regulatory elements can also lead to human genetic disease. And we have another big problem with this, that unlike genes where we have this code that we know that 3 codons, basically three nucleotides code for a certain amino acid, we don't really know the code of the regulatory elements. And so what my lab has been working throughout the years is using high throughput approaches to really understand the code and how if you change it, that could lead to human genetic disease. And we've also been trying to find mutations in it that lead to human disease. And more relevant for today's talk is we've been trying to use regulatory elements to fix genetic disease, because they're the switches that turn on and off genes. And so there's a lot of genetic diseases where you have the genes that's expressed at lower levels or at higher levels. And so if we can mess with the regulatory elements and change them, for example, to reduce expression levels or to increase expression levels of a gene, we might be able to cure some of these diseases. And I'll show you the first types of diseases that we started with in the lab, and those are called haploinsufficient diseases, and this was initiated by an extremely talented postdoc in my lab, Navneet Matharu, who's now an assistant professor also at UCSF, and also a CSO of a company, Regal Therapeutics, that we formed for this, that is trying to do this for many other diseases. So the first classes of gene dosage diseases is what's called haploinsufficient diseases. It's kind of a long name, but it's actually, if you break it out, it's quite easy to understand. So haplo is basically half, and insufficient, just not enough. So half is not enough. And what does that mean? So we usually have 2 copies of a gene, one from mom, one from dad, and that leads to normal gene levels. So we have a full gas tank of proteins that are expressed. But every so often, we could have a mutation in one copy that leads to a loss of function, and that copy now is not working. And so what we have is basically 50% of RNA being made and then 50% of protein being made. And surprisingly, this is fine for many genes, and Angelman syndrome is an example for that, where UB3A, normally you get only 50%, and that's fine, right? But there are, and we went through the literature, 660 genes that actually, when you have half, that leads to human disease. And the diseases could be devastating, including epilepsy, cancer, polycystic kidney disease, and many, many others. And the thought is that the numbers are even much higher. So there's these, now that we have sequencing that's much cheaper, we can sequence a lot of genomes, and we can look in what genes we never see a loss of function mutation, a mutation when one copy is not working. And people showed that over 3,000 genes might not have this. Of course, some of these could be lethal, and that's why we don't see them. But in terms of disease, and we went through the literature quite a lot, we see that there's 660 basically genes, diseases that are caused by this. So how can we fix this? So I think you've all heard about CRISPR, which can come to the DNA, to a certain location, and cut the DNA and potentially change it. But what I'm going to talk today about is a different type of CRISPR, and that's called CRISPRa. And so the idea here is that you take the CRISPR molecule and you actually make a mutation in it, so it cannot cut the DNA. What that does is, 1, it doesn't cause any changes to the DNA, so it's sort of a bit safer. But 2 is that you can use it as a delivery truck to bring things to a specific location in the genome. And for what location, for example, you could use it is, let's say, a promoter or enhancer, these regulatory elements that I mentioned before, active regulatory elements. And I can bring to it, for example, is something that will force them to work harder, to just make more RNA from that gene if I bring it to that specific location, something like an activator. And that would make much more RNA from that gene, and as I said, that's been called CRISPR activation, CRISPRa for short. So how does that tie to the haploinsufficient model that I mentioned before? You have still in this model one existing copy that's perfectly fine. It's just giving you 50%. So what we thought when we heard about this approach is maybe we could use it to squeeze out more RNA and more protein from that existing copy, just force it instead of making 50% to make 100% from that single copy, and that will increase the amount of protein being made and could fix this disease. So when we started this, we wanted something quantitative, something that we can measure, that even if we increase a little bit, we will see some kind of an effect. And so we chose obesity as the model to look at, because all you do is you weigh the mouse in this case and see, can you rescue that phenotype? And so we chose 2 of the top obesity genes that lead to this phenotype. And when you have one copy of them, you have mice that are extremely obese, and you have humans that are extremely obese. Usually, they die at age 50 from cardiovascular issues, diabetes, and are bedridden for a lot of their lives. And to make this short, picture always shows it nicely. It worked, and you could see here on the left a mouse that has one copy of a gene called SIM1 that is extremely obese, and same holds for human. And in the middle, you have a mouse that has exactly the same genetics, the same genotype. It just has one copy of this gene, SIM1, but you could see that it looks much more like the mouse on the right than the mouse on the left, meaning that it's actually pretty normal due to this approach. So this is the most complicated slide in my talk, and I'll try to go through it slowly, but the main gist of this is just to show you also the advantage, and I'll show you 3 advantages sort of this approach. One is that it's very specific in terms of where it functions, and this is the main sort of takeaway from this slide if you don't understand. It has a big advantage because we're using the regulatory machinery of the genes themselves. We're not sticking in a lot of AAV or a lot of genes in there, but actually taking advantage of the own regulatory machinery. And so, for example, for this gene that I just showed you, SIM1 is expressed in the hypothalamus, in the brain, and also in the kidney, but it's not expressed in other tissues, and I'm just showing you as an example lung and liver. So where you have dark red here, that means that it's expressed. When they did this experiment, we actually made this in a transgenic manner, meaning that the CRISPR-A machinery was expressed in all the tissues in the body, and this is just showing you 4 tissues, but we looked at all, and you could see that the CRISPR machinery is expressed in the hypothalamus, in the kidney, in the lung, in the liver. So it's expressed everywhere. So technically, we could be upregulating this gene in all tissues because we have the CRISPR machinery there targeting it and trying to upregulate it in all tissues. So we wanted to see, are we doing that? And so we looked at the expression of this gene in various tissues. Again, I'm just showing you 4, and you can see this is just showing you normal mice, wild type, and you can see that we plot that as 1, and we see expression in the hypothalamus and in the kidney, but what BDL means below detectable levels, we don't see it in the lung and the liver. And if we look at the mice that have just 1 copy of this gene, we could see that they have 50%, as you can see here, for the hypothalamus and the kidney in orange. So what happens if we upregulate this gene, and again, we have the CRISPR machinery in all the cells, and you can see that we're actually just upregulating in the hypothalamus and the kidney, but not in the lung and the liver when we target the promoter of this gene. Even though, again, as I said, the CRISPR machinery is everywhere, in every tissue. And in this experiment, we also targeted an enhancer of this gene that was far away from the gene itself that's very specific to the hypothalamus, to the brain. And what you see is that we only upregulate in the hypothalamus, but not, for example, in the kidney or anywhere else. So it's very specific to what we target, meaning that if we target any one of these active regulatory elements, it will only upregulate there, but not upregulate in other tissues. And so the regulatory element that we target provides us tissue specificity. And so it's not just expressing everywhere, or upregulating everywhere, but just based on the regulatory element that we target. Another way that we approach this is with adeno-associated virus, which you'll hear a lot about, which is used a lot in gene therapy. And another advantage we felt that there is for this technique is length. And so adeno-associated virus is great for gene therapy, but it can only package 4,700 nucleotides in that virus. And so that could be a big problem for large genes. And here I'm showing you going back to these 660 genes that lead to haploinsufficiency in orange, or the 3,000 that are predicted. And if I draw this red line that you see of what would fit into an AAV, and when you give an AAV, you also need to have other things in the AAV in addition to the gene, like a promoter to turn it on, this active regulatory element. You need other stuff to make an AAV. And so it's a bit even smaller. It's 3.8, I would say, is what you can fit. So you can see that on the right, there's a lot of genes that actually I can't package into an AAV. And so using this gene replacement approach of putting that into basically to rescue when you have a missing gene will not work actually with AAV, but potentially our approach with this CRISPRa, by just upregulating a gene, could work because it could fit into an AAV. And the last advantage of this approach is when we use AAV, it can, in particular in neurons that don't divide, it can stay there for the life of the organism, and we think it could provide a long-term therapy. And so what Navneet did here was inject into the mice this virus into the hypothalamus, exactly where this gene is expressed. And what you could see here on the left is that when she injects, you do get levels similar to normal. So in orange on the left is what you get from in a normal mouse. And the second in orange, and you can see, is half in a heterozygous mouse that has just one copy of this gene. And in blue is a mouse that's a control where we inject just the activator, but we don't tell it where to go into the genome. We don't have a guide, and you can see also in blue that we're not getting to normal levels. But now when we tell it to go to the promoter in yellow or in green to the enhancer, you can see that now we're getting levels that are similar, of this gene, similar to normal mice. So the next is, can we rescue their phenotype? So we did this at 4 weeks of age. We inject into the mouse. We wait 7 weeks. And you can see that after 7 weeks, this is how much food they eat and how much body weight they gain. And you can see that the yellow and the green look very similar to the one on the left, the normal. So they don't eat as much as other mice, suggesting that we can rescue their phenotype. And more important, in terms of long-term therapy, we took these mice all the way to 9 months of age. And what I'm showing you on the y-axis is how much they weigh after 9 months. And so a normal mouse weighs about 30 grams after 9 months. You can see that these heterozygous mice that have just one copy of the gene, they weigh over 50 grams. They're quite big. Our control in blue, where we don't tell the CRISPR machinery where to bind to, but we have it in there, you can see also weighs similar, so we're not rescuing. But when we have a guide that tells it to now bind to the promoter of this gene or to the enhancer, you can see that they weigh 30 grams even after 9 months from that single injection at 4 weeks of age. And so they have a normal body weight, basically, at that time. And we do see that the gene is upregulated still, and that the CRISPR machinery is still active at that 9 months of age, suggesting that this could be a long-term therapy for this. So all of that was published a while ago, and I'll show you a bit more of what we've been doing since then. And so one gene that we set out also to target for this is SCN2A, and that's thanks to families of SCN2A and the Salmons Foundation, and John, who's here. And so the work that I'll show you was done by an extremely talented graduate student in my lab, Serena Tamura, and Perry Sprott, who's a graduate student in Kevin Bender's lab, who's in red in the middle. And this is also along with Stephen Saunders, who now recently moved to Oxford from UCSF. And so what is SCN2A? Well, John gave a beautiful introduction of the amazing work that the Simons Foundation did in characterizing all these autism-related genes. And this is actually the newest characterization. And when you look at all of these genes that we didn't know before could lead to autism, the one that stands out at the top is SCN2A. It's actually the most causative gene for autism. And it leads to very severe autism, developmental delay, intellectual disability, and others. And so what is it? It's a sodium channel, and it's very important early in life in neuronal excitation. But then later on, after 1 year in humans or 1 week in mice, a different gene takes over called SCN8A, but SCN2A has this important role in backpropagation, sort of really controlling basically neurological function. So we did the same approach here for SCN2A. We target the promoter, so the regulatory element right next to the gene of SCN2A, to see if we can upregulate it. And we do this in mice in one side of the brain, so we can use the other side as a control to see does that work. We wait for 4 weeks for the virus to work, and then we take out the brains, we measure are we upregulating, and we do a lot of electrophysiology on the neurons to see are we rescuing the phenotype. And so I'm also not a neuroscientist, so don't worry if you don't get this. The main thing to see here is that in blue is the mice that have one copy of this gene. And you can see that the peak here in the bottom row is much smaller. But when we upregulate in green this gene, you can see that we're rescuing this. And we could look at a lot of mice, a lot of neurons, and see that we can rescue the electrophysiological defects. Another key thing, and what was really exciting, both in terms of Angelman syndrome with the ASO and what John showed before about Rett, is that we find here, also in mice even, that we can rescue the phenotype even in adolescent mice. So these mice are actually P30 when we inject. They're actually much bigger. And everything that I'm showing you here is actually a rescue in mice that are almost at adolescent age. And so that was extremely exciting for us, in particular because we looked at their neurons. And when we look at them at that age, or even later on, you can see, again, if you look at these peaks, and AMPA and MDA is sort of a measure of how mature their neurons is, you can see that they're much less mature in blue here because you have this higher peak. But when we rescue, you can see that we're getting them again to normal levels. And again, we're injecting in mice that are at age 30, so 30 days of age. So they're really adolescent and much bigger. And we can also measure that. We can look at their neurons specifically, and you don't need to really know what this is. And as I said, I'm not a neuroscientist also. I'm not great at this. But what you definitely see is that we are rescuing, and we measure this across many mice, many neurons. We are rescuing the phenotype, and the pink here looks much more like the gray, suggesting that we are getting them looking like wild-type mice, like normal mice, using this approach, and that we can rescue this. We also went here to human cells. So everything I showed you before was mice, but we went to human cells, iPSCs, or stem cells that have just one copy of SCN2A. And what we do here is we differentiate them for 3 months in a dish to make them into neurons. And then after 3 months, we either give them CRISPRa to upregulate the gene, the existing copy, or we just give them, like before, the CRISPR machinery, but we don't tell it where to bind. And you can see here in the left in black is the normal cells. In the middle are the cells that have one copy that we just give the CRISPR machinery, but it doesn't know where to bind. And you can see that there's no basically electrical recording hardly from these neurons. But when we do let them know where to bind to the SCN2A and raise the expression levels of that existing copy to make it instead of 50%, 100%, you can see that we can completely rescue the phenotype, and you see basically neuronal waves there on the right in purple, suggesting that this approach could be used also for other diseases. And again, we're extremely excited here that we could do this also on adolescents, showing that the brain is probably much more plastic than we think, at least in terms of this gene. So that brings me to Angelman syndrome and what we're hoping to do for Angelman syndrome in terms of this project that we're starting. So as Allison nicely pointed out, UB3A is the main culprit, and there's beautiful work being done now to basically try to target UB3A. However, as Allison also mentioned, 70 to 80% of the patients have a deletion. And so in that deletion, there's at least 16 genes that will be missing in terms of having one copy. And looking into the literature, we think that at least 5 of them could be haploinsufficient, or it could lead to some sort of phenotype because they have just one copy. Similar to everything I've showed you before for a single gene, there could be an issue with having just these genes having one copy. So what we hope to do is try to upregulate these genes using our approach, all 5 of them together, basically in a single manner, to see if upregulating all 5 of these might be beneficial and help in terms of improving the phenotype. And so what we're doing, or starting to do, is develop first a one guide, so we're trying to learn how to upregulate each gene separately, one by one by one. And we're optimizing now techniques in the lab as we speak to upregulate all of these genes individually. And then once we do that, to get this closer to a therapy, we're developing a virus that can upregulate all 5 together. And so that way, we could upregulate all 5 of these genes in a single sort of experiment in a single shot. We're extremely lucky and fortunate, thanks to Allison, to be put together with Albert Kong and North Carolina State University, who's developed these beautiful organoid models where he can also rescue UBE3A, and they have the whole entire deletion. And so what we can see is what happens when you rescue UBE3A, what phenotypes do they have, and then we're going to send our viruses to Albert to see, can he rescue the phenotypes in these mice when UBE3A is rescued? So is the upregulation of these 5 genes simultaneously going to be helpful in terms of the phenotype in these organoids? And then, of course, mouse models are extremely important. And again, thanks to Allison, we're working with the Jiang lab in Yale, who developed a similar mouse model that is missing all of these genes. And their same thing is we're going to generate the same thing, so via guides that target all 5 genes, but this is in mice, so the sequence is quite different. We have to develop them sort of separately from the human. And then when we develop them, we're going to make virus and again ship it to Yale so they can test in their mouse model, can we rescue some of these phenotypes in the mice? And so we hope that this could potentially be an approach that could basically help with the remaining genes in this region and other than UBE3A. So with that, I'll end with the most important slide of thanking my lab mates, which are amazing, and none of this could be done without them. Our collaborators, in particular in UCSF, Kevin Bender and Stefan Saunders, and Christian Vasa for the obesity work that I showed you, and our new collaborators, Albert and the Jiang lab at Yale, and which we're really looking forward to work with. And our funding, it could not have been done without Families for SCN2A, the Simons Foundation, and now with FAST. And happy to take questions, I think, if I have time. Thank you so much.
Are these CRISPR treatments a one-time treatment?
Yes, supposedly, they could be a one-time treatment. There are concerns with CRISPR that is important to know is that immunogenicity, we don't know how the body will react to CRISPR, and a lot of companies are working on that. But supposedly, as I've shown you in the obesity mouse model, we did a one-time injection in 4 weeks, and we carried them up to 9 months, and they were still fine. The issue, again, is once you insert something, it's there forever, so you want to make sure that it's safe.
So in terms of getting to a therapy to humans, would you have to deliver into the brain, or how would you do that?
Yeah, so in terms of delivery, it would depend on, the way we've done it is AAV delivery into the brain. There's been beautiful work by the Jiang lab for basically other ways with nanoparticles or lipids that could be done, or other things. So we focus on just showing as a proof of concept that this could be done. Once it is, the delivery could be in any way that you deliver DNA. So it doesn't have to be with AAV, it could be with other things that could deliver it.
Hi, I have 2 questions that sort of blend into one. First question is piggybacking off the delivery. If you decide to go the AAV route, would you have to deliver 2 things then? Because opposed to the mouse where you can just make them have the DCAS9, you have to put that in the human. So are you thinking having 2 different AAVs, one for DCAS and one for the guides? And then the second question is thinking about trying to rescue the multiple genes in the deletion genotype. Are you thinking about the possibility of maybe using a different enzyme that allows you to have like an array of guides targeting the promoter region of these different genes? Or are you thinking about having each guide in a separate package and then co-delivering like 6 AAVs into the brain? Thank you.
Yeah, great question and really important question. So we're doing this as a proof of concept to show that it works, but at the same time, we really want to get this to work as one virus if we can. And There's been a lot of advancements in the CRISPR technologies. There's very new Cases, like Cas12, for example, which will be ideal for this, where we could basically package, it's a much smaller sort of CRISPR that can fit into an AAV, along with the activator, and along with 5 guides. And so that's, I think, what we're leaning to. How it'll be in the clinic in the end, this field changes so quickly that it's hard to know. But definitely, I think that's really important, because the more viruses you have, and that's what you're getting at, if we do this with 5 viruses, it's not going to be as effective than if we do it with 1, because we'll need to get all 5 into a certain cell, and that's hard. But 1 is much easier.
Great. Why don't we wait for the rest of the questions for the panel so we can stay on time? So just reserve them, write them down, and we'll have all the time in the world to ask them on the panel. Thank you.
Thank you.